Good evening. So I'll start with some scary charts. Usually when you talk about AI, people think about the scary world where AI is going to take over. Now I'm going to show you the scary world where AI does not take over. And the problem is, if you look at the chart at the top, is the cost it takes to make new discoveries. So for drugs, it's pretty clear, actually. There is this law that's called the E-room law. If someone can figure out why it's called E-room, because it's Moore law reversed, is the fact that the return on investment for drug discovery is exponentially decreasing. And there are some economists who actually did some funky analysis that kind of show that because the number of researchers is almost increasing exponentially, but our growth rate actually is kind of almost decreasing, it may be true not just for drugs, but overall for humanity, that the cost of discovery is increasing exponentially. So the challenge is what to do about it, and that's where benevolent AI comes in. So our view is that computers have an edge over scientists. So when you talk about AI, you always think about, oh, okay, how are we going to make a system that's better than people at the tasks that people do? But the great thing about science is that there is a lot of data that's relevant to science. So if you take the field that interests us most at the moment, which is bioscience, there's a huge amount of data around diseases, around molecules, around genes, and it's impossible for a scientist to analyze all that data. So think about it, there's like a few thousand to like 10,000 papers that are written every day in bioscience. And scientists at best can read a few weeks to really understand them, or maybe a few days if you want to be nice. So there's a huge amount of data out there, a lot of it actually unstructured. And what you need to do is can you make a computer that can read all this, not necessarily as well as a scientist, we're not trying to replace the human here, but to a point that it can get some interesting insight from all that information that can help a scientist make new discovery. And this is kind of the foundation of the company. So if you look out there, there's 90 million full-text patents, 30 million scientific articles, a little bit of structured data that actually has been extracted sometimes out of these papers. And can you create a system that actually can analyze all of that and put it in a format that actually come up with interesting hypotheses, interesting prediction, or at least interesting starting point for a scientist to make new discovery. And so that scientists with their advanced brain can actually make new discovery with access to all that information. So what I'll do today is kind of give you a quick view of how we do that. Before I do that, I'll tell you a little bit about the company. So we're three and a half years old. We've raised a good amount of money. We're actually really two companies put together. So we're not only just doing AI and software engineering, we're actually doing also drug development. So we actually have scientists really in the company doing drug discovery, and whose goal is to actually, we actually started just now a clinical trial phase 2. So we're going all the way from the AI to the actual clinical trials. And it's an interesting, I would say, social experiment, because you're putting together people from very different backgrounds. I come from a tech background. I'm a kind of AI guy. I've been in this space for 20 years. And you have people who have been in drug discovery in actually sometimes large companies like Pfizer and the Merck of the world, trying to put them together in one company, really work together. And what we use, we use squads coming from Spotify, actually. So one of our model comes from really the tech industry to make them together. It's actually a very interesting experiment to do that. And so that's our mandate, to use AI to accelerate scientific discovery, and we are starting with drug discovery. So what I'd like to do actually is to show you what it means in more concrete terms. What is the kind of analysis we do. Our process really uses 4 steps. The first step is we go out there and we collect all the relevant information. Actually, completely non-trivial work to do that. There are actually a lot of unstructured information sources I mentioned, like patents and clinical trials and publication especially. But there's also a lot of actually structured knowledge bases, especially in bioscience. People have spent a lot of time collecting some amount of structured data, like there's a data source called Campbell, another one called KEGG about pathways. But all of them are very incomplete. All of them are very limited, and they are not very consistent. There are different points of view on that data, and they're also very much not exhaustive. They represent, I would say, 5% of the knowledge. So the first step is, go out there and collect all that information and put it and process it to make it a usable format. From that usable format, you can have the scientists explore the data, or you can get the system actually really explore the data to make new hypotheses. Basically come up, if you think about it as, if you have a disease, come up with new target for that disease or new mechanism for that disease and propose them to a scientist. And that's really the trademark of what we do, is that instead of thinking of discovery as a process where you start from scratch, we have a system that always generates hypotheses so that the scientists start from a list of something, with some prediction of how good they are, okay? And so that's the second step of the process. Based on all that information we process, come up with new hypotheses. If the scientist gives you a disease, give them some mechanism and that are druggable so that they can actually start from there. The third piece is that as we give them hypotheses, they will actually try to validate them, and we'll try to say, okay, are these good hypotheses or not? And we track and we see what they do as they do that, and they give us a lot of feedback through this validation. And as you can imagine, as we get this validation, we can use a feedback loop to improve our models so that next time we show a hypothesis, it actually takes into account that feedback. And the third piece, this is actually a whole process that's called repurposing, because everything we do here is based on existing knowledge. So we are trying to find existing molecules and find new use for these molecules. But we also have a whole application of machine learning to chemistry as well. So at some point we find an interesting molecule for a disease, but we may find that that molecule actually has some imperfection. For example, it cannot pass what's called a blood-brain barrier, okay? And in this case, you want to modify that molecule to come up with a new molecule that's actually more satisfactory. And for that, we use actually machine learning algorithm that now using this generative network can actually come up with new molecules that are modified. And again, the scientists, the chemists would actually look at a list of molecules rather than come up with a molecule themselves. So again, the same principle of coming with new hypotheses that the scientists will validate. Let me go through the few steps. So the first one, as I mentioned, is take all this structure and the unstructured data with this, understanding that the majority of the information actually is in unstructured data, and convert it into a format that a machine can understand, and also humans can understand. And you can think of that as a really large, knowledge graph. Think of it as like 1 billion facts, okay? And each of these facts is actually motivated by some information that we can highlight into all that unstructured information. To do that, we use multiple steps. The first step is, as I mentioned earlier, actually a fairly large, I would call semi-automated curation work. We have scientists who are actually a bioinformatician. We have around a dozen of them in the organization. They go around, collect all these sources, and write some intelligent scripts to try to combine all that structured information into something that's actually consistent and relevant. The next step is, if we stop there, it's not enough. You need to use some machine learning that would actually use that as a foundation to come up with an understanding of the text. And for that, we use some interesting techniques. I would say, if you're doing, well, maybe some of us have more chance, but a lot of new startup, I would say, these days, can't afford to spend a lot of time training and creating training sets for supervision. So I would say the sweet spot these days is what we call weak supervision or semi-supervision for startup. And that's what we do here. So think of it as our problem is trying to actually understand the language in bioscience papers or patents or publications. And the challenge is we're not going to get some people to annotate that information. The traditional way to do that is to get someone to actually read the papers and highlight the key entities that matters and the key relationship between these entities, and you will learn actually from there. But instead what we do is we use what's called distance supervision, because we have access to actually a lot of structured information that I mentioned. It's only 5%, but it gives you an example of like a relationship, like this gene or this mechanism is relevant for that disease. And it gives you an idea that if you find that the name of that disease and the name of that gene in a sentence, there's a good weak indication that actually it's a meaningful relationship. And so we use this kind of, bootstrap the system by using all the structured information that's very limited, but gives you a piece of information to understand actually the language that's in the document. There are a lot of advantages to do that, rather than using, very supervised sets that people will actually annotate, is because, every month, I would say, we are trying to refine our understanding of relationships, for example, in our data sets. So you can think of, gene A being relevant for disease B, but you could also think of gene A inhibiting B, or gene A actually triggering B, and you can look at or gene A triggering B in tissue C, and so you can have very much more refined types of relationship. And if we had a fixed graph or a fixed set of training information, that would actually be pretty limiting and pretty expensive to maintain. So that's our first step. Go out there, collect all that information, leverage the structured information to bootstrap the unstructured information parsing, and then create a large, very much, if you want, knowledge graph with some evidence behind it. That's a system that we actually make accessible to our scientists. We also build a lot of application on top of that so that they can navigate the system. But more interestingly, we use that for our own system to generate new hypotheses. And so the main interface of our system is you really go, you enter a disease, okay, and the system will actually give you a set of mechanisms behind the disease and a set of a sub-network of network biology, if you want, behind it. So, we will come up with some evidence because, scientists won't just take the computer for, the computer suggestion for granted. They will want to know, okay, what's behind it. And so then the system try to actually come up with evidence behind the hypothesis came up with, usually through a different, actually, algorithm. The interesting thing about this is that we can try to use signal from many, many different places. So we use a lot of ensemble techniques that use information that comes from the structured, from the unstructured, that use different strategy to come up with new hypotheses for a given disease, okay? And we present that to the user and we get feedback from them. Now the third piece, as I mentioned, is we're not at the state today where you put a disease in the machine and press a button and you get a pill at the end. The drug discovery process is still like a 10-year process. We're trying to make it much faster. But the scientist is still in the loop, right? So the system is coming up with a hypothesis and the scientist will go through them. It's actually, interestingly, something we're reconsidering right now. We're thinking sometime maybe you want to actually take that information and put it into an assay directly, test it into a cell or an animal, but that's something we're just still talking about. Right now, the scientists actually validate the hypothesis. What they do to validate it, they will go deeper. They will look actually into the evidence that the system provides for that hypothesis. Usually, it's a set of, you can think of it as a bit of some network biology behind the mechanism. Gene A, is related to gene B, that's related to gene C, that's known to be related to that disease, okay? And each of these relationships will have actually a set of evidence behind it, so the scientists can click on them, see the passages directly, the sentences highlighted in the paper, so they can actually have access to a very large number of papers very quickly through the interface that actually supports a given hypothesis. So they can go deeper and they can go also wider. A big problem is to understand what's actually novel. I mean, understanding and trying to quantify or qualify even talking with scientists what's novel is quite complicated. And so the scientists will try to figure out, is the hypothesis actually novel? Are there patterns around it? So we give them access to, again, huge amount of information. So very quickly, they can validate this hypothesis compared to the known knowledge out there, okay? And the nice thing about this is that as they validate the hypothesis, we try to create interfaces that everywhere in the system we'll try to collect feedback from them. And from mundane problem, sometimes actually, we make a mistake and understand, I mean, if you have done a little bit of life sciences, you'll see that there's dozens of ways of calling anything, and sometimes you have the same way of calling two different things, and you need to differentiate all this, so the system will make quite a lot of mistakes on this, and we're constantly trying to improve this, disambiguate actually the terminology, to the most advanced, which is, okay, we give them actually a hypothesis of a mechanism, they have to tell us if they thought it was an interesting one or a not interesting one, or they don't really know. And obviously the key from this is to always create a kind of a feedback loop so that, as they evaluate the system, we feed that back into the model, I would say the most actually interesting things we have done in the past 6 months is to apply the same model to also chemistry. And there's a bit less related on text, but you start looking at molecules and really create a system. I mean, the innovation we're bringing in is to create a system that not only can predict that's something that pharma has done in a while, trying to use machine learning. Here in our case, we try to use more advanced machine learning algorithms like deep nets to try to predict properties of molecules. Do you have like protein-protein interactions? Is this chemical a skin irritant? Can it pass the blood-brain barrier? It's actually quite complicated and expensive to do experiments to measure that. So if you can get a system that actually measures it in silico with some level of accuracy, it's better. But what's even more interesting is once you can predict this thing is to create a system that comes up with new molecules, right? So you tell it, oh, okay, you start from that molecule, but actually this thing doesn't pass the blood-brain barrier. Can you suggest new molecules that have the same kind of good properties, but not the bad properties? And we have just come up with a new system recently. I think we'll publish something and we'll submit for publication next 2 weeks. And we are at a point where we can come up with new molecules that actually chemists don't laugh at. Now, there are lots of great papers that have been written around using machine learning and neural network around generating new chemistry. But if you really look at them, if you actually get chemists to look at what's generated, they will say, that's a joke, okay? They're not, they look good on paper, when you really look at them, they don't. So now we're at a point where we actually can generate molecules that scientists are like, oh, actually, yes, that's a reasonable molecule, but look, it's actually super toxic, and I would have never picked it, okay? So that's where we are right now. But still, our belief is that you can create a system where, by coming with, hypothesis, I have one more slide, but for the scientist, you can really accelerate that process. I just have one slide. So the last slide actually is if you ask, does it work and where are you right now? So we do have actually good evidence that you can find some signal in the literature and through the chemistry using these techniques. And we have actually kind of a very interesting story around Lou Gehrig's disease, ALS. That's the ice bucket challenge. This is one of the most complex diseases out there, very, very difficult. So we are not specialists. So we have around 15 drug discovery scientists in the organization, and they're not specialists in the field. But we basically, gave our system to someone who is a good life science person, has done the drug before, but not in ALS. And they were able to actually, maybe it's me touching this, or they were able to actually generate some very interesting hypotheses in just a few weeks for ALS, 5 of them. And then usually we go see some specialists in the field, so people who have been there for 20 years. And they looked at the 5, and they kind of cut them in 2 categories. Half of them look very, very, something they could have come up with. Actually, I think there's one of the 5 that a guy there actually had a really good idea and he thought he would actually do. And the other 2 were completely off the chart. They thought they were very unusual hypotheses. And so working with them, we actually put the 5 hypotheses in the lab. At this point, it's in vitro and in vivo validation. And 2 of them didn't work well. One of them worked as well as this kind of standard of care at the moment. And 2 of them work actually much better. And even one of the scientists said it was one of the most interesting discoveries he's seen in his lifetime. He was kind of young. He was like in his 30s, I think. So there is some good evidence that you can accelerate the process here. But I think we're still at the beginning of the journey. And our vision is that if we can do that for drugs, we may do that also for materials. We are actually looking at battery technology, solar cells, and things like that, using the same kind of exploratory techniques. That's my talk. Thank you.
Thank you very much. Fascinating. So this sounds like it's quite extensible to other things. Do you or can you also cover physicians' handwritten notes, for example, to inform discovery?
So first on the extensible, it's not that it's really easy, right? Just think of it right now. We have just 15 bioinformaticians just focused on drug understanding of life science data for drug discovery. So if you want to reproduce that in another domain, you will have to actually also have a lot of bring in domain knowledge and also bring the scientists. But we believe that the principle could be applied, especially the molecular exploration like this. And today, to answer your second question, no, we're not using at patient data at the moment. There's something you could do around drug discovery, but it's not the primary data source.
All right, questions?
Hi, and thank you. If I'm Pfizer, should I be scared? Like, are you driving me out of business?
No, because, in some way, we're not really competing. It's a big space, the space of discoveries, right? And we're not competing directly. But there is this thinking really in the industry, and it doesn't come from me. I'm new to this industry, right? I've been in it for just a year. that large pharma is not really the research engine anymore, and that a lot of the large pharma actually will acquire more than develop themselves. So there's this kind of thinking that they're very good at commercializing and marketing, but in terms of developing new drugs, maybe they're not the best place to do it. And there are a lot of reasons for that. Some of them very cultural, more than, and some of them also in terms of incentive. If you're in a large pharma and you're just in one area, your goal is to push your programs to the end, even if it's not going to work. Okay, and so countering this kind of incentive is very important to be successful, because you want to fail fast and early. And in pharma, that hasn't been the case.
In medical research, the publications are done only up to a certain level of probability of certainty. So in other words, approximately 5% of all findings that are published are incorrect. What does your system do to eliminate these inconsistencies and problems in the data? So
we don't, if you give us a paper, we can tell you, oh, that paper is really well written and the data is perfect for it. It's quite a difficult problem. Actually, we're thinking about it to give you a rating. But I would say the main tool we have for that is aggregation mass, right? So when you look at a piece of evidence in our system, you never look at it in isolation. You'll always look at it with all the experiments that have been done in that area, and you can collate it. But there is some thinking we're having, and we're talking with people as well, that you could try to use that to try to predict, and a bit like what people were saying before, predict how likely a paper is to be reproduced at some point or not, right? So you could start doing this kind of stuff, but I don't think it will be very, very accurate, but you can start looking at these things. But so you have to use all this, and our scientists are human like all, and they come up with new hypotheses, and they come up with their own bias as well. And this is why we're starting to think sometimes that should we even, for many things, try to bypass the scientists and go directly into in vitro and do the experiment there and backtrack to evaluate how good our scientists are, and see that. So there's some things that could be done, actually.
I'm sure they would love that.
Well, it's an interesting cultural problem, actually. We're trying to actually hire a scientist who wouldn't mind, actually.
Very interesting. All right.
So how do your scientists actually pick their project? And do they have any economic considerations with it?
That's a very good question. So we have a priority. So as you must have understood, we actually are very disease agnostic, and we're trying to stay disease agnostic, but then we have a prioritized list of disease, okay? And we're not, at this point, I feel like it's too much, it's still too much expertise-based rather than data-driven in terms of that list. And one of the factors is commercial viability of the system. And so we're looking at that, but I would say we're still at a point that's a bit ad hoc. This is part of me trying to transform the company, so we're really, really data-driven everything. But it's a factor. So very early on, you're trying to see, like, if it's a disease that has been looked at 1 million times, and that's very hard, and the assays are very hard, and the in vivo model are bad, and it costs $100 million to do a clinical trial, and we're not going to try to look at it.
Great. Maybe last two, so one for Adam here, and then.
You spoke a bit about the challenges of having a model and then having to try and come up with a model to explain to the end users why the model made those selections. Can you talk about how your users reacted to that and what some of the challenges with that were?
Yeah, I mean, it's a common problem. So we're not necessarily telling them that we're using different algorithms for both, but I think that's one actually of the trick, right? I mean, you can always transform. In my view, I think it's a bit of an overblown problem, because you can always transform a predictive algorithm in 2 steps. Whether your first step is trying to create, the supporting evidence for your prediction, and so it's kind of a bit of a adversarial network where you try to summarize information as much, and then you try to basically still predict based on summarized information. So you can always use these techniques to give you supporting information. Now, what it means is that also your path is not exactly the same. So if you don't have that constraint, you can make a little bit better hypothesis. So usually what you do is you make the hypothesis, and then you try to justify them, and you don't tell your scientists that's what you do here.
Okay, so last question. preferably somebody who hasn't asked a question before. There was one here. Thanks, Andrew.
Thank you. So you talk about the target-driven drug discovery approach. It's usually very difficult to come up with the target itself because that's closely tied to our knowledge about human biology. And I want see whether you could elaborate more about how you guys build this knowledge graph to help scientists know better about ourselves, better about biology, sort of fasten the learning process of how much we know about human bodies.
So all our learning obviously comes from existing knowledge, right? It's in the literature. So really you can think of it as trying to process all the literature for it. We don't come up with new targets that have never been looked at. For example, the system by definition cannot do that. So it's always about repurposing some existing knowledge. And if you think of target, for example, one pattern we're trying to find is, the simplest, so you have a target-target interaction or a protein-protein interaction or a gene-gene, and then you have a gene-disease interaction. And maybe, target 1 is target 1 to target 2 is well known, target 2 to disease is well known, but target 1 to target 2 disease is not well known. And there you can have an edge that shows you something novel, but people have never put that into that context. So it's just a very simple example, although it's something actually we do use. So we kind of jump hoops, and realize that actually nobody else had done that jump.
Great. Well, thank you very much. This was terrific. We appreciate it.